we're running a little bit behind. I'd like to introduce you now to Sarah Miller and Niveen Ismail. I don't know if I pronounced that right, Niveen, from Smith College, and they will talk about best management practices.
Hello, everyone. I am Sarah Miller. I am a junior in the engineering department at Smith College, and today I will be presenting research from Professor Niveen Ismail's lab, Best Management Practices, the application of mycoremediation to reduce E. coli in runoff. So best management practice is a practice that inhibits or removes pollution from a non-point source of pollution. We're focusing on agricultural runoff as the non-point source of pollution, which can introduce bacteria, pesticides, and other contaminants to local bodies of water. And to manage the agricultural runoff, we are focusing on a bioretention basin, which is a type of best management practice that uses filtration methods enhanced with vegetation. For our bioretention basin, we are using mushroom mycelium as our vegetation type. And you can see on in this image on the right, an example of a bioretention basin in practice. So during a storm event, you can see that the bioretention basin is flooded with runoff, and then even just 24 hours later, that runoff has been filtered and the basin is now clear. And then on the left, you can see a cross-section of a bioretention basin where the runoff flows through the pipe, over the rocks, and then through the plants, through the sedimentary layers that filters the water, and then it flows out through the base. So we have seen that fungal mycelium attacks bacteria by releasing antibiotics and toxins, and it has even been shown to use bacteria as a source of nitrogen and carbon, particularly in nutrient deficient environments. Certain species of fungi have even been capable of degrading harmful toxins and bacteria by stimulating microbial communities. So this is a timeline of this research. It began as course-based research in an upper level engineering elective course at Smith College with support by Phil Crosby of Fungi Ally. And you can see that it has gone through many different iterations. We began with testing different filtration methods of this runoff. So we have coffee filters, steel filters, brass filters, and we also have used different types of systems. So we have stacked nalgenes, we have some bags full of mycelium, as well as our column designs with packed mycelium. And many students over the years have contributed their time and hard work towards this project. For our bioretention basin, we chose to use pearl oyster mushroom mycelium because we know that it is highly popular among mushroom growers in the Northeast region. These mushroom growers have a high interest in finding usage for the spent substrate blocks, which is left over after growing mushrooms, because it is otherwise just a waste stream. And we also know that pearl oyster mushrooms are highly popular in the marketplace. We also have seen that previous research on the capacities of pearl oyster mycelium finds that it's excellent at removing and filtering microbial pollutants, specifically E. coli, from water. And you can see on in the image, this is one of our mycelium blocks. It is pearl oyster mycelium, and it's inoculated on sawdust. Our first objective for this research was to test the functionality of our newly designed packed columns. And you can see in the image that we have a transparent PVC body with fitted endings. There is a fitted inlet for the hoses that are in the carboys filled with the synthetic stormwater that allows them to flow through the columns. And then there is an inlet in the base that allows the water to exit the column after being filtered. We are also testing to determine the efficacy of using mycelium to remove E. coli from synthetic stormwater, specifically over multiple storm events. And in this image, you can see that 3 of these columns are mycelium-packed columns, which are experimental columns. One of the columns is a control column, and then we have 2 of our 20-liter carboys, which are filled with spiked synthetic stormwater. So one of our controls, the first control, is the control carboy that contains the synthetic stormwater spiked with E. coli, and that has the same concentration of E. coli as what flows through the columns in the experiments. And then our second is the control column that is identical in cross-section to the experimental columns, with the exception that the 15 centimeter layer has sawdust instead of the 15 centimeters of packed mycelium. And in this cross-section of the design of the columns here, you can see that the E. coli flows through the top, down through the different layers, and out through the bottom. So we have the first layer that is 15 centimeters of pearl oyster mycelium inoculated on spent sawdust, 10 centimeters of soil, and 5 centimeters of fine-grained sand. Our initial E. coli spike amount was 10 to the 6th colony forming units per 1 milliliter of sample, with a flow rate of 1 liter per hour for 6 hours. Each of the experiments we conducted included 3 to 4 storm events, where a storm event lasts 24 hours. 6 of those hours are the wetting period, where the water is flowing through the columns, and that is followed by an 18-hour drying period in which there is no water flowing through the columns. In between each of our experiments, we had a drying period of about 5 to 8 days in which we did not flow any water through the columns. In this top image, you can see our sample processing setup. So we use serial dilutions to dilute the samples, and then we did membrane filtration, and then we placed the filters with the E. coli onto the MTEC plates here, which were then incubated for 24 hours. This bottom image is one of our MTEC plates with the filter with the E. coli colonies. Each of the purple dots represents 1 E. coli colony. From this, we are able to count how many E. coli colonies are present on the filter, and we can calculate the concentration in the filter or in the sample. So here's my first graph. This is from our first experiment. And on the x-axis here, you can see we have storm events. So for this first experiment, we had 3 storm events total. And then on the y-axis, we have C over C naught, which is the concentration in the sample divided by the initial concentration of E. coli colonies in the control. The blue bars on the graph are our controls. So the light blue is the control carboy, and the dark blue is the control column containing sawdust. And then the 3 shades of green are each one of our mycelium-packed columns. And for this first experiment, we saw approximately 1 log of additional removal by the E. coli, by the mycelium columns. And this is really what we're looking for when we're researching microbial inactivation, because this 1 log of removal indicates that there is a change in the concentration, and that's just a factor of difference between the experimental columns and the controls. For this second graph, we have our second experiment. So again, the first 3 storm events on this base are from the first, and then we had a drying period of about 5 to 8 days before we began our second experiment, which had storm events 4 through 6. And you can see that in the 4th through the 6th storm events, there was an increase in removal by the mycelium packed columns, which are the green bars again. And there's little change in the controls at this point. And then for this final graph, we have our third experiment, which are storm events 7 through 10. And we had about 5 to 8 days of a drying period between the second experiment and the third experiment. And in this third experiment, you can see that there is a decrease in removal by the E. coli colonies. As you can see, the green bars on the graph are decreasing in length over time. And we did observe an increase in removal by the control column containing sawdust, and that is the blue bar on the graph. We also noticed after looking at these 3 experiments that the last storm event of each experiment, so storm events 3, 6, and 10, that there is a decrease in performance by the mycelium packed columns. And we hypothesize that this is caused by a higher saturation in the columns at this point in each experiment, which is reset by the drying periods in between the experiments. So for the first 7 storm events, we observed approximately 1 log of additional removal of E. coli by the mycelium packed columns as compared to the controls in the experiment. However, in the remainder of the storm events, which are storm events 8 through 10, we did not see more removal of E. coli by the mycelium columns than the controls. In addition to this, we also saw the control column containing the sawdust had an increase of E. coli removal over time, which we hypothesized was caused by a growth in the sawdust at some point in the experimental process, which resulted in the additional inactivation of E. coli. We did not test the sawdust in time to identify what that growth might have been. However, from previous research, sawdust should not be removing E. coli from synthetic stormwater. We also saw that there was very little mycelium growth in the test columns after the first couple days in the experiment, particularly in storm events 1 through 2, we observed some mycelium growth. However, after that, there was very little to no mycelium growth observed. And we hypothesize that this also may have contributed to a decline in the efficacy of the mycelium packed columns to remove E. coli from the synthetic stormwater. In this image, you can see that there is a red circle around this top of this layer, and that those white spores in that circle are what we look for to see mycelium growth. That's this mycelium growth. And you can see it's very little just at the very top of the layer. But we would expect it to extend throughout the layer over time as the mycelium continues to grow. However, we did not observe that. In the future, we plan to design a new system that better suits mycelium cultivation by creating more optimal growing conditions for the mycelium, as well as to better mimic field conditions that the bioretention basin we are designing would be implemented. We also will continue to investigate the efficacy of using mycelium to remove E. coli from synthetic stormwater long term. We hope to use this research on mycelium to implement bioretention basins to reduce the input of E. coli from agricultural runoff into local bodies of water. We hope to improve the quality of water in these local bodies of water by preventing that E. coli from flowing into local bodies of water from agricultural runoff. We also hope to partner with local mushroom growers to find beneficial usage for the spent mushroom blocks because they take a lot of time and energy for mushroom growers to manage, as well as a lot of space to compost the blocks. And if we can find a beneficial use for these mycelium blocks, we would be creating a circular economy for the blocks to find a better usage. I would like to acknowledge that this work is being funded by Smith College Picker Fellowship. We would like to thank from Smith College, Mark Anderson, Sue Froelich, Dale Renfro, Eric Jensen, and Gabby Immerman for project support. I would also like to acknowledge the contributions and past work of all past students, including Ojaswi Aryal, Tiana Cooley, Ruth Pemberthy, and Sophia Garcia Carlin. And we would especially like to thank Mycotera Farm and Fungi Ally for their support and contributions of mycelium. And I would like to open the floor for any questions and comments.
That was an excellent presentation. If you can't think of your questions right now, please put them in the